Hi, everyone. Welcome to our Tuesday seminars on the cerebellum. Today we have Chiara Cirellli from University of Wisconsin, who is going to tell us about the work on effective sleep and how it promotes homeostatic plasticity, synaptic pruning. And I'm so grateful to have this opportunity to listen to her. Chiara, thank you so much for your time with us.
Thank you. Thank you very much for this opportunity. As I mentioned before, the Cerebellum is our latest study, but is part of a more general effort to try to understand how sleep affects different parts of the brain. And so you will see, I will discuss the cerebellar study eventually, but I want to start by giving you a broader overview of what we do. So first of all, what I'm going to tell you today is all done in collaboration with Giulio Tononi, my long-term collaborator. And over the last 20 plus years, this work is done in Madison, Wisconsin, at the Wisconsin Institute for Sleep and Consciousness, and is funded by NIH and by the Department of Defense, and I have no conflict of interest to declare. So this is my overview of what I'm going to tell you about. First, just a very general brief point of why we think we sleep researchers, of course we are biased, but we think sleep is very important, and why we think so, and then some ideas about the functions of sleep, and then I'll go and discuss this hypothesis about synaptic homeostasis. So why sleep is important? Well, there are at least two fundamental pieces of evidence. First of all, if you look around, all animal species that have been carefully studied so far do sleep. And you see here some examples. There are more than 230 species studied so far. So of course there are still few, but basically all the ones that have been studied until now are found to sleep. And you see some examples like the latest addition to the list, like the Cassiopeia here, or the flatworm, or of course a little bit older examples, the fruit fly or the sea snail, Aplysia californica. So that's one reason we see sleep in all species. The second important reason is that sleep is very tightly regulated. So there are very complex mechanisms in the brain that allow you, if you lose sleep, let's say you have one night without sleep, the next day you will sleep longer and or deeper, meaning your sleep will be more consolidated with less brief awakenings, with more slow waves. And I think this is a very strong evidence. You know, some people still, a few people still believe in the null hypothesis, that basically sleep is kind of a time filler, that we sleep when we have nothing better to do. However, it's very difficult to account for the presence of these homeostatic mechanisms if you think the sleep is just a time filler. Why would you have this regulation? Now, but if it's important, what is the function of functions that we think sleep is doing? And in one sense, you can all say, well, we all very well know why we sleep. If we don't, we feel miserable. Next day, everything is so difficult to achieve, which is absolutely the case. But more specifically, we know certainly that sleep is important for many body functions. So I'm not here to, of course, reject the idea, because there is a lot of evidence that sleep is important for metabolism, for immune function. But what I'm going to tell you about is mostly about the beneficial effects of sleep on the brain, because there is compelling evidence that most reliably, if you lose sleep, even in the short term, the impairment is at the cognitive level. And it's very difficult actually to find an aspect of cognition that is not affected by sleep loss. So you see here some examples, studies, attention, working memory, ability to learn, to consolidate memory, ability to integrate a lot of information, all these, even humor appreciation has been studied. So all these cognitive aspects are affected by sleep deprivation. And the last one, which is kind of a important new area of research in our field, is this effort allocation. So even when you actually test people after sleep deprivation and you find that in some tasks, their performance is within the normal range, but they will very often tell you that it was so difficult, so much more effort that was needed to get to that level of performance. So that's the evidence. Definitely, if we lose sleep, cognition is broadly impaired. And also, very importantly, I'm not going to describe to you these studies, but this is just to tell you that we also have evidence that once you have this impairment due to sleep loss, it's only by sleeping that you can recover from this impairment. So just having instead a period of quiet wake or rest in which you close your eyes, but you are still awake, doesn't do it. And here are some examples of, in different tasks, visual tasks, or more motor rotation adaptation tasks, in which this has been carefully studied. So we need the sleep to recover, but why is that the case? So again, what is exactly in the brain that we need that can happen during sleep? And so here is, we have always thought that if you want to try to address this question, you have to start from the fundamental feature that is distinguishing, that defines sleep, that distinguishes sleep from any other behavioral state, including, as I say, just quiet wake, which is, as shown here in this beautiful painting by Rousseau, partial sensory motor disconnection. So by definition, when you are asleep, you are less able to respond promptly to potentially a very dangerous situation. And so we think that you can have any idea about what sleep does for the brain, but you have to account for this, because if the function, the essential function of sleep could be performed during wake, during quiet wake, evolution would have found a way to just do so, because it would be so less, potentially dangerous. And we think that the function, basically, that can only be done properly when the brain is offline is indeed to maintain synaptic homeostasis. And so that's the idea that I'm going to tell you about now. So, of course, you know very well, synapses are at the basis of how the brain works. There are many, many of them, 100 trillion or so just in the human neocortex. And most of them look like this. So these are excitatory axospinous synapses, because that's basically the majority of the synapses, 80% or so in the mammalian brain. And that's what I'm going mostly to tell you about. So synapses are at the basis of how the brain works, and we know very well that synaptic activity overall accounts for a big chunk of the brain's energy budget, 70% or so. There are different estimates, but everybody agrees that that is a major component. So obviously everybody agrees also that you need to keep synaptic activity very tightly regulated because of the energy costs. So here is a general description of this hypothesis, synaptic homeostasis hypothesis of Shy for short. So that basically during the day, the 16 hours or so for us humans, we are always learning something new. is not just when you, hopefully today you remember what I'm telling you, so you go to a lecture or a class or some formal form of learning. Learning means that your brain adapts to an ever-changing environment, and that is happening every time you are awake and you move around and you adapt to the environment. So there is always ongoing learning. And because of that, at the end of the day, we think there is a net increase in synaptic strength in many brain circuits, which is indicated here by these red lines all over the brain. Because of that, this is good because it's a sign of plasticity and learning, but basically the job of sleep would then be to overall produce a net decrease in synaptic strength, to rebalance this change during wake, to make sure that then the next day, basically, you start anew overall with the same level of synaptic strength all over the brain. Now, of course, and I will discuss this briefly at the end, this process cannot be just a specific global downscaling. If that were the case, of course, everything that you learn during the day basically would be lost. It has to be a process that is broad, but it has to be specific. And so that's why we call it down selection. And I'm happy to discuss how we think this happens at the end. But basically, so that's the points of the rationale for these hypotheses that I don't have time to go in great details through them, but they are especially discussed in this review in 2014. So basically what I told you, learning must occur when we are confronting the environment. So during wake, it happens mainly through potentiation, but that of course creates immediately the need for a process of renormalization. And we think this renormalization happens primarily during sleep. And it is because of this process that actually we can account for all the beneficial effects of sleep at the system level, the ones that I mentioned before for cognition, as well as, of course, at the cellular level in terms of saving energy. So learning happens through potentiation mainly, not only, but mainly, and here are several of the studies that have, formally tested this idea. I don't think this is controversial. It's also not controversial that, again, you need a process of renormalization. Here I'm just showing one example, but there are, as you probably know better than I, dozens of studies with computer simulations in cortical networks, hippocampal networks, showing that if you implement the rules of plasticity, of strengthening, then definitely you immediately need also rules of renormalization. Otherwise, you will have runaway potentiation, saturation of synapses, and basically epileptic activity. So everybody agrees on this, but many people, I think, still think in the field that the brain is smart enough to be able to maintain this balance at any given time. So let's say, you are learning because you are now strengthening some connections and potentiating some connections because of your exposure to my experience. But at the same time, there are other connections that are weakened so that the balance is kept. While we think, again, that this is not the best way to allow renormalization, we think that this process should happen during sleep. So basically, during wake, you have mainly net increase in synaptic potentiation. This morning, I go out, I see many things. Definitely the brain is able to, in the short run, to form many, many memory traces, helped fundamentally by the high levels of activity of all the arousal systems in the brain. Noradrenaline here is key and paramount, but is not the only one. So the activity of the noradrenergic system, dopaminergic, cholinergic, orexinergic systems, histaminergic system, all these arousal systems are more active during wake than during sleep. And by virtue of being more active, they bias plasticity towards potentiation rather than depression. Then we go to sleep. And here is the opposite. These systems have low activity. For the noradrenergic system, serotoninergic especially, they get totally silent in some parts of sleep. And that biases the plasticity on the opposite side towards weakening. But crucially, and we go back to what I say at the beginning, we are disconnected from the environment, and yet the brain is very active. This is probably the single most important finding in our field over the last century. The brain doesn't stop firing during sleep. It fires. It's just that the pattern of activity is different. And for a large part of our mammalian sleep hours, there are slow waves. There is synchronous activity on and off of most neurons, of almost all neurons, for instance, in the thalamocortical system. This condition, we think, is key for the brain to sample all synaptic weights and then produce this process of smart down selection. Then again, seems kind of magic right now, but I hope by the end of the lecture, I will be able to explain. But again, the crucial here is that this process of sampling and down selection happens while we are no longer slave of the here and now of the environment, because we are disconnected. And therefore, we can implement the down selection without having to adapt to the environment. And that's the key reason why we think sleep is the perfect time to produce this renormalization. Now, very briefly, of course, the main claim of this hypothesis is that there must be, if you sample synaptic strength overall in the brain, in many brain circuits, at the end of the wake day, there must be a net increase in synaptic strength relative to when you sample here at the end of sleep. Because if this is not true, then the entire hypothesis, of course, is not correct. And so we have spent a long time, over the last 20 years actually, to try to measure synaptic strength in many different ways. Unfortunately for us, there isn't a single measure, especially if you study animals, in vivo, freely moving animals, there isn't a single measure of synaptic strength. There are molecular, electrophysiological, ultrastructural measures, as you will see. But so our approach has been to try to use them all. Some are better than others, but basically to try to use them all and see whether at the end we get a consistent picture. And to do this as much as possible across species. Literally, we have done it from flies to humans, because again, if we believe that this function, maintaining synaptic homeostasis, is a key function of sleep, it has to be true across species, not just in us or not just in a mouse. So electrophysiological markers, that's what you can do. You can stimulate, of course, from neuron A and record the monosynaptic response in neuron B. Classically, this, you can measure the amplitude of the response or the slope. So we have done that. We have done it in mice and rats through electrical stimulation, but you can do it non-invasively in people using TMS. You give a small pulse, you do exactly the same stimulation over different times of day. So here the wake duration increases and you see the slope also increases. So this is a night of sleep deprivation. After that, the slope is even steeper. And then after a night of recovery sleep, then it goes down. Now, this is one evidence. I'm showing this as first evidence because it's the least compelling, because there could be many other reasons why the slope, the evoke responses changes due to changing intrinsic excitability that are not directly related to synaptic strength. So another way that is more direct is by using the minis. So you take the brain, you take a slice here, for instance, of the cortex in a mouse, in a rat, after several hours of wake, or after several hours of sleep. And then what you measure are these events, spontaneous events that reflect the spontaneous release of glutamate. And people agree that the numbers or the frequency of these events and the amplitude of these events is actually a direct measure of synaptic strength. And you see here that definitely both measures are increased after short deprivation, or also is not shown here, but the same results happen after spontaneous wake at night in a mouse or in a rat because they are nocturnal animals. Now, I don't want to explain this, but this is just to say that, you know, after we published, this was published in 2011, I think, but then there have been several new studies from other labs testing, measuring these minis, and basically they all find in different cortical areas results that are consistent with our original result. Now, what about molecular markers? Those are, I think, stronger evidence simply because these markers for the glutamatergic excitatory synapses, especially in mammals, are extremely well established. But we have used also in flies, actually, other markers, presynaptic markers like this BRP. So basically, the concept is that, well, if this synapse is strong or gets stronger, the total amount of proteins, either pre or postsynaptic proteins, must increase. And this is what we see, for instance, here is in flies, and that's, I think, the only study that I'm going to show you in flies. But if you take the entire fly brain, as you see here, this is after a fly. Flies like us are actually diurnal animals, so they sleep during the night. So here is a fly that slept all night. The expression of this BRP, that is a very ubiquitous presynaptic protein, is quite low. Instead, after a night of sleep deprivation, there is overall increase in the expression of this protein. Now, in mammals, the best markers are on the postsynaptic side. They are these, the expression of these AMPA glutamatergic receptors, because there are literally hundreds of studies showing that after synaptic potentiation and strengthening, the total amount, basically, of these receptors, especially the GLUA1 containing receptors, or their phosphorylation levels, increase, and vice versa. When you have synaptic depression, the expression of this marker decreases. And so that's why we use them, because they are very accepted markers. And so here is our original study, by now quite old, in which we took the entire half of the cerebral cortex in a rat after 6-7 hours of wake, again, either spontaneous wake or sleep deprivation, or here, the hippocampal formation, and after wake or after 6-7 hours of sleep. And then you extract from the synapses with this synaptoneurosome preparation these proteins, and you run a Western blot to quantify these proteins. And you see that overall there is a 30-40% increase in these markers, many of them, after wake relative to sleep. And this, on the right, this is just to say that this is another study that was done more recently in Rick Ugenir's lab. They used the mice rather than rats. They took the entire forebrain, so basically most of the brain. And for them here, 100% is the level of all these markers after wake at night. And relative to that, what you see is, again, a decrease in many of these markers after 3-4 hours of sleep during the day. So I think the molecular evidence is quite compelling and is quite consistent across labs. And again, very importantly, these differences are seen when you sample large brain regions. It's not just one specific synapse, because I would argue that even if you see this change, but only in a very small circuit, that's then hard to understand that that would be the function of sleep. But more recently, we also thought, meaning in the last 6-7 years, that perhaps one of the most stringent ways of testing these hypotheses was by literally measuring the size of synapses. because fortunately for us that there is a very good, strong correlation between function and structure. So it should be the case that if we are right and overall synapses get stronger after wake, they should also be bigger, and vice versa. They should literally shrink after sleep. Of course, the synapses are small, so we need electron microscopy and serial electron microscopy to test this, and that's what we have been doing. This is our microscope. This is a serial block phase scanning electron microscope that we have in the lab and literally runs day and night, basically. And here you see an example of one of these image stacks that we can collect. This was one of our first studies. So this is the cortex, the motor cortex of a mouse. The methodology, I don't know if you're familiar with this, but basically is almost automatic, meaning you have your piece of tissue inside the microscope, and there is a diamond blade here. So it cuts automatically under vacuum. It cuts a very thin section, 50 nanometers usually. Then it takes the image with the scanning electron microscope, and then it cuts again, it keeps going. And in our case, you can get, again, 500, 600 of these slices and images overnight. Then the problem starts. The problem meaning this is relatively quick, but then all the reconstruction of what you want, we want, which is, of course, the synapses, the spines, and their size, still has to be done. We still do this manually, and that's what is extremely time-consuming, because we want to reconstruct in our tissue, cortex or cerebellum, all the dendrites that we see in the stack, and all the spines here, the axonal boutons. And what we really care about actually is this red area here, which is the direct area of contact between the pre-synapse, so the axonal bouton, and the spine head, which is the post-synapse. Because this axon-spine interface is a very good measure, ultrastructural measure of synaptic strength. So it's basically this red line here that, of course, it becomes an area in 3D. We could have used other measures because they're all very strongly correlated to each other. So for instance, you can measure the volume of the spine head, or you measure the postsynaptic density, or actually you can count even the vesicles containing glutamate. All these structural markers are strongly correlated to each other. We chose the ASI because for us in our images, that's the most reliable measure that we can get, in which we can really very precisely track and segment this area. But as I say, again, there are a lot of studies, classical electron microscopy studies, showing that all these measures are strongly correlated to each other. So it doesn't really matter which one you choose. And most importantly, that they are related to functional measures. For instance, here our synaptic area is correlated to the number of these AMPA receptors that are expressed in the synapse. And actually then these receptors are correlated with the amplitude of the currents mediated by these receptors. So to make a very long story short, because this is published since a few years, we have done first cortex and the hippocampal formation, so primary motor and sensory cortex, and then the CA1 stratum radiatum. Now, in each of these studies, we were able to reconstruct fully almost 7,000 plus of these spines and synapses, and we got quite similar results that I'm going briefly to describe. But I want to go back to this point that is very important. When we do these studies, like when I described to you, I told you about the synaptoneurosomes, for instance, I mentioned sleep deprivation or spontaneous wake. We always use these two conditions. So we have two wake conditions and then the sleep condition. sleep, rats, mice are nocturnal, so they sleep during the day. So you want to sample or take the brain at this time here. Let's say 3 p.m., which means after 6-7 hours of sleep. Spontaneous wake is 3 a.m., after the animal has been awake for several hours at night. But if you only have these two conditions, when you compare them, the animals are differing not just in behavioral state, but also in time of day. And we know there is a lot of evidence that in the brain, gene expression and many functions are just changing because of circadian time. So we have to be very careful in correcting, controlling for that. And that's why we use this extend wake or sleep deprivation. So this is an animal that is killed at the same time of day as the sleeping animal, but is kept awake because we literally play with the animals. They are used to play with us. We give them novel objects. So when I show you results of sleep versus wake, is always when there is a finding of sleep versus both wake conditions to control, again, for circadian time, as well as for possibly stress or aspecific effects due to the stimulation of the sleep deprivation. So here is the first study in which we have these 3 conditions and 4 mice per condition, which is the cerebral cortex, as I mentioned. We sampled both the motor cortex and the sensory cortex, but the results were basically identical. So we pooled the data. And here, what you see, each of these little dots is one of these ASI, one of these areas here, which is our measure of synaptic strength. And you see here, overall, the distribution of the ASI size in the sleep group is down relative to the 2 wake conditions. And here are the same data shown in this cumulative graph. You see that the sleep curve is shifted to the left relative to the other 2. And in fact, at least at the population level, we could test that is a downscaling. It's not global because it's downscaling because on average, all synapses change by 18% in proportional to their original size. It's not global because the very top here, the 20% or so, the largest spines, like these mushroom spines, they're called, they do not seem to change between sleep and wake. We speculate that this is because these are young, but already kind of mature mice, and those synapses are already carrying important memories that don't simply change between sleep and wake or after a few hours of sleep deprivation. But of course, this is a speculation. Now, just want to mention that we have done also a similar study, again, with electron microscopy and focusing on the primary motor cortex, the superficial layers. That's where we studied this in very young animals. So these are 2-week-old pups. Now, of course, at that age, like in newborns, you don't have long hours of spontaneous wake at night. So we can only have 2 conditions, sleep for 3-4 hours and sleep deprivation for 3-4 hours, which is extremely difficult to enforce, of course. But the take-home message here is that even at this time, developmental time, indeed, we see a difference in the ASI size and therefore in the strength of synapses. Actually, on average, is larger, is 34% mean difference rather than 18%. And at this level, at P13, actually, it seems global scaling. And we think perhaps it's because this time is still very early on. Animals are still learning. Some of these pups don't even have eyes open. So all synapses are still completely plastic and they can change between sleep and wake. But this is basically to say that, you know, it was our assumption that if this process of maintaining homeostasis is very important, it has to happen also early on in development when sleep is so prevalent. And at least for the motor cortex, it seems that this is the case. Now, I won't go into the details, this is published, but basically we have done in the same animals, so literally physically, these are the same animals in which we studied the cortex, and now then we took image stacks from this area, specifically the CA3 to CA1 synapses, and there, overall, the results are the same, so that we see in sleep versus the wake conditions a difference, a decrease in size. In fact, in the hippocampus, for the largest group of synapses that are this group here, we even see a difference in the number of synapses. So the number decreases after sleep relative to sleep deprivation. But what about the cerebellum? Of course, for us, the cerebellum was very interesting, not only because it seems that everything that we study in the cerebellum seems to behave in a different way relative to the forebrain, but also, of course, because the cerebellum houses the largest number of synapses in the brain. And specifically, of course, those are the synapses established by the granule cells through the parallel fibers to the dendrites of the Purkinje cells. So that's what we study. And we focused on the posterior vermis in lobule 6. One reason being that, you know, it's extremely important because at the end you are going to study and focus and measure, and it takes, I forgot to mention, but every one of these studies requires then manual segmentation, requires 4 or 5 people working full time for at least 3 or 4 years. So it's extremely time consuming. You need to be careful and you need to be sure that you are sampling roughly the same region exactly across animals. So you need some reference points to help you. And this region was easy for us to identify. We are in the molecular layer, in the most distal part of the molecular layer, because there were previous studies showing that that's the part in which almost all, if not all, synapses in the spines come from the parallel fibers, not from the climbing fibers. And so here again, here you see an example of one of these image stacks, again, 500, 600 slices, 50 nanometers each. Overall, we were able to segment more than 7,000 of these spines. And because almost all of them harbor one synapse, overall, we have more than 6,800 synapses. Here, these are literally all the pieces of dendrites that we were able to reconstruct in the 3 groups of mice. And again, these are again the same mice from which then we published the cortex and the hippocampus. And this is, I think, very interesting. First of all, again, we can compare directly. On the right here, you see an example of a dendrite of a Purkinje cell that we reconstructed. And you see relative to a representative dendrite in the cortex, the cerebellum is very, very crowded. Of course, this was known before, but for us, it's impressive to see the difference. Not only that, but you see that in cortex, again, the size can vary quite a lot. There are so-called thin spines, very small, or these huge mushroom spines. And overall, the distribution of the size of the spines and synapses, which we measure here as the size of the ASI, is log normal, which was described before. In cerebellum, here is almost unimodal. So there aren't basically large spines. So they are all kind of small, medium, and all very much homogeneous. Now, first big difference, indeed, we find again that the cerebellum behaves differently. We don't see a difference in the mean size of the ASI between sleep and wake conditions. But we see a difference instead in the proportion of spines that do carry a synapse. So I told you before that, you know, as actually, this is true also in cortex and in hippocampus, basically in an adult animal, almost all spines have a synapse. in the cerebellum specifically is 93% of them across all animals. And they look like this here. So they are kind of club shaped. And here is the ASI. So the ASI, the synapse is established on one of the side, is not on the tip of the spine head. This was also described before. Sometimes this is, most of these spines, 85% of so, are single spines. But there are 15% are so-called branched. So there is one neck that then splits in two. And so there are two spine heads, and each of them has a synapse. The 7% that are lacking the synapse, we call them naked. They look like this, for instance. Or it can be branched with one side lacking a synapse. Now, what we found, again, is that is the proportion between these and these, between the spines that have a synapse and those that do not, that changes between in the cerebellum between sleep and wake. So here in the center, you see a representative example of a dendrite after several hours of sleep. And the blue ones are the spines, the naked spines. Their proportion is much higher, is twice actually, than the ones in the two wake conditions. So basically, after sleep, 10% of all spines are naked, and on average, only 5% are naked after wake. So this is very surprising, was very surprising for us, because it meant that, again, while in the cortex and in the hippocampus, sleep and wake seem to be able to modify the size of the synapses by 18–20% on average, but it's not the synapses appear or disappear, while in cerebellum, the changes seem to be much more clear-cut, all or none. Either a synapse is present or not. And we also found, and that's also extremely interesting, I would be really looking forward to learn from you what this means. This change in being able to have a synapse and then missing a synapse does not happen on any kind of spine. I told you most of the spines are single spines. It actually happens mainly in these branched spines. Those are the ones that overall can either acquire or lose a synapse. And so the absolute number or the relative number of these branched spines is double, is 16% after wake and only 8% after sleep. So far, now we are doing a fourth region that is the striatum, but as I told you, it will take a few years before I'm able to tell you about that. But basically, for the three regions that we have studied so far, we see that overall the effects of sleep is in the same direction, but each region has quite distinct way of doing this process, it seems, of renormalization. So in the cortex, it is all about decreasing the size of the synapse of the ASI. In hippocampus is a mixture between size and number. In the cerebellum, it really seems to be all about these all or none changes in the number of synapses. And one interesting aspect in the cerebellum was that, when we study these branched and we reconstruct them, synapses, and then we can follow, of course, the parallel fibers establishing contact with them. And what we see is that in almost all cases, more than 90%, 93%, I think, cases, these are different parallel fibers, which may have, functional consequences. Now, I will finish by saying, well, I hope I convinced you that we have quite some evidence about the overall main claim of these hypotheses, that, there is a net increase in synaptic strength after wake and a net decrease after sleep. But I also said at the beginning that we think this can account for the beneficial effects of sleep at the cellular level as well as at the system level. And why is that? Well, at the cellular level, it's probably quite easy to understand. Of course, if you have overall weaker, smaller synapses, you need less energy, less supplies, proteins to sustain synaptic activity. And of course, you're also able, you move these synapses away from saturation, which is good for, future learning. And so indeed, you can see, and these are, again, I'm not going to describe this, but there are several studies proving that indeed the sleep helps the next day to be able to learn and encode new material. And again, this may be not so difficult to understand in terms of, well, if you, do synaptic renormalization, you are in a better position for new learning. The most perhaps difficult aspect of this hypothesis for many people has been to try to understand how can it be that overall synaptic weakening, which is what I've shown you, can account for the fact that actually sleep helps in memory consolidation, because many people equate memory consolidation with synaptic strengthening. We know there is a lot of evidence that many tasks, declarative tasks, as well as procedural tasks, benefit from sleep. Not all of them, and we still don't understand that. That's a very interesting aspect, actually, in sleep research, why some tasks do not seem to benefit. But overall, the evidence is compelling that sleep helps in memory consolidation. And so there are these two major ideas. One thing is, sleep can help by doing this process of down selection, of renormalization, but it could also at the same time do what is called the active systems consolidation, in which some synapses, perhaps related more specifically to what we have learned that day, get actually strengthened in absolute terms during sleep. Trying to tease this apart, trying to see whether these two processes can happen together, or one is happening more than another in different conditions, is very, very difficult, because of course you need sleep, you need a freely moving animal, but at the same time, you need to be able to follow synapses, single synapses. So there is very little known, but this is our first attempt to try to do so. So we are using this complex wheel task. The mouse learns in 1 hour to be able to stay on the wheel, although the wheel, keeps running at faster and faster rates. And basically, we have done this 3-4 times. It's very consistent. After 1 hour, one session, animals get better. Then you let them either sleep or you sleep deprive them for 6-7 hours. Then when you test them at the beginning of the second session, if they slept, they start at a higher level. So they maintain basically the performance that they had at the end of session 1. Instead, if they're sleep deprived, they start lower. So sleep consolidates this motor task. Now, what we have done has been, finally, with 2-photo repeated imaging to try to follow what happens to the synapses when the animals is learning this task. So we have these mice in which, they are genetically engineered, so we can look at the dendrites in primary motor cortex and the spines. And so we have a structural marker, as well as CEP-GLUE1 is one of those molecular markers for the AMPA receptors that I mentioned before. And if you quantify the expression of this, it's a measure, it's quite a direct measure, actually, molecular measure of synaptic strength. So in this study, we can literally, with 2-photo repeated imaging, look at the spines and quantify them before sleep, after sleep, in normal conditions, no learning. Then the next day, immediately after training in this task, and then we either let the animal go to sleep or we sleep deprived, and we image it again and we image again. It's very difficult, of course, because you have to be able to go back exactly to the same spines. But basically, this is what we see at the end. We have been able to, measure 1,500 of these spines. And what you see here, this normalized difference is basically the overall synaptic strength in all these spines. And not surprisingly, I've shown you evidence for this. At the end of sleep, normal sleep, overall, there is a decrease in this marker of synaptic strength. Also, not surprisingly, the next day, immediately after the task, there is a net increase in synaptic strength because, again, it was known before this motor task is mediated through potentiation of synapses in motor cortex. Then also, not surprisingly, we see that, if the animal is not allowed to sleep, overall synaptic strength remains at this level. And instead, if they can sleep post-learning, it goes down. But what was very, of course, new for us and we wanted to know was not just overall all synapses. We wanted to study specifically what we call the max synapses, so the ones that really were potentiated after training relative to all the other ones. Because this increase here that you see after motor task is driven by a minority of the synapses. It's only 10% that show the largest increase in synaptic strength. Everybody else changes very little. So we are asking, well, after learning occurs, what happens to the max spines relative to the other ones? And here is basically what happens. So these are the max spines by definition, are the ones that they increase the most after training. And quite surprisingly, we found, I'm plotting here them separately for the two groups. After training, it doesn't really matter whether the animal sleeps or is sleep deprived. In both cases, even 24 hours later, they still are quite strong. They weaken a little bit, but not much. And again, it doesn't matter whether there was a sleep or sleep deprivation. The difference is all driven by the other spines, the ones that did not change much with the motor task. And again, those are the ones, they are 90% of all spines, do not change if you are sleep deprived, but they go down with sleep. And so now, if we express the strength of the max spines as relative strength, so relative to all the other spines, you see that at the end of sleep, of course, the max or learned spines in sleeping animals are in a better position than in a sleep-deprived animal, simply because everybody else went down with sleep, but not with sleep deprivation. And we also found that, the extent to which the other spines go down with sleep positively correlates here with the performance. So the more there is down selection of the other spines after sleep, the better is the performance of the animal. So this is just to say, so in this, of course, is just one task. It would be nice to be able to measure this in other tasks. In this particular case, sleep increases the signal to noise ratio, not because it strengthens in absolute terms the max spines, but simply because it allows the weakening of all the other spines. And so in this case, we can say, the memory consolidation, as well as, of course, the ability to avoid saturation and the energy saving can all be accounted by the renormalization. You don't need to invoke absolute strengthening. And so that's basically my talk. I just want to mention, of course, at the beginning, I say it seems down selection is kind of magic. What we know at the molecular, there must be a specific selective way in which you produce this weakening in the synapses. There are many studies done in our lab, but many other labs now that are basically identifying molecular tags that can then help some synapses to stay at the level of the max synapses to escape the down selection and others not. So there are positive or negative tags, because at the end, if you want to weaken one of these synapses, these glutamatergic synapses, the only way you can do that is by the endocytosis of these AMPA receptors. And we know a lot about the different molecular markers. The other very important point that now we have learned is that the way at the electrophysiological level, the way synapses can escape, or neurons in general can escape the down selection, is by continuing to be very active during sleep. If you are active, then this process of down selection does not happen, again, because you tag the synapses. I'm happy to discuss, this will take, describing this will take 1 hour or more just in itself, but I just want to mention, now the field has moved mainly to understand exactly how this process can be smart and selective in producing this renormalization. Thank you.
Thank you so much. Let me begin the questions for that. So really lovely, what a beautiful presentation. So thinking about the cerebellum and imagining how this might work with regard to the hypothesis in terms of pruning, the purpose of making the system more energetically efficient, we think of the cerebellum as a location where learning largely produces depression of the synapses. So what do you see the results that you have with regard to the different learning modes that exist in the cerebral cortex versus the cerebellum?
Yeah, so I think my reading, that was one of the starting point for us of doing this study. The general idea that also we studied that, in the cerebellum, the main form of plasticity is through synaptic depression. But my reading of the literature now is that this view has been quite changed over the last, at least 10 years. That in fact, if you look at all the major synapses in the cerebellum, including, of course, the parallel fiber to Purkinje cells, in fact, all forms of plasticity are present. And for some forms of learning, vestibular learning or not, actually seems that the dominant forms of plasticity, like everywhere else, is not potentiation, is not the depression, but is potentiation. So that's my reading of the overall, more recent literature, that this view of how the cerebellum is different was very much biased by studying one specific condition of co-activation of parallel fibers and climbing fibers, but in only one condition, and that doesn't translate to more general forms of plasticity. So again, it was the starting point of our study, but the more I read the literature, the more I realized that from that point of view, it doesn't seem like the cerebellum is very different from any other area.
I think somebody have raised their hand. Vincenzo, you have raised your hand?
Actually, my question was just done by Reza, but I want to follow up on this, because indeed now we have some learning paradigm for which we think that potentiation is more important, and others for which we think that depression is important. But in view of these differences, what is your current hypothesis now on the cerebellum? Like, do you think that the synapses that potentiate during learning during the day for certain tasks, then they potentiate during the night, while the one that are depressed because of learning, then they potentiate during the night, potentiate back during the night?
Yeah, again, it might change depending on the exact task, but overall, so that would be my, my prediction. Now, you have to understand that these animals, these are animals that are doing what I mentioned as ongoing learning. They are not learning any specific task. Certainly, they are exploring, they have a running wheel, so they move around, but in our condition, for this study, in this study, we didn't employ the complex wheel task and test them immediately after learning. So this form of very basic plasticity that we see between sleep and wake is something that seems to be going on every day, independent of the specific motor task or any kind of task, actually. So to answer your question, we will have to, carefully adapt several tasks and then see at the synaptic level exactly what changes and whether it mimics what we find in these conditions or not. For us, in general, the surprising part, the most surprising part, at least for me, was this issue that there seems to be this really all or none change, because losing in a few hours, because again, we are comparing, not even after a task, 6-7 hours of sleep with 6-7 hours of wake. And that seems to be enough for literally synapses and ASI in our measure to disappear or reappear. That was the surprising thing. And that's because, and then reading the literature, our possible explanation that we have in the discussion of the paper was, well, maybe that's because, you start in the cerebellum with many, many synapses, however, small synapses with small, weak synapses that supposedly, at least according to some studies, are so weak that actually are silent. They don't make any difference at the cell body when you try to recruit them. And so they are there, but actually they can be moved around, appear, disappear quite rapidly. And the other fundamental point was that this seems to happen in this very specific type of spines that are the branched. Now, branch spines are present in the same overall proportion of 10-15%, also in cortex and hippocampus. But there, they don't change between sleep and wake. Here, that's where this losing or acquiring a synapse happens. And perhaps, again, this is very speculation, but we were trying, based on the literature and the data, computer simulation and modeling data that are available, to make sense of this in terms of perhaps those are what people have called the eloquent spines. Those are the ones that have a chance to make a difference. If there are at least, people have counted the 120,000 or up to 175,000 contacts between parallel fibers and dendrites. Then maybe, as shown recently, confirming previous studies, only 50% of those are actually real contacts, but still a huge number of synapses. How can a single synapse hope to make a difference to the cell body and being able to drive and fire a Purkinje cells? If you are a branched spine, so you actually have two synapses coming from two parallel fibers, and maybe those two fibers get activated simultaneously, they have a better chance of making a difference and be eloquent. But of course, this is all speculation and is driven by these very unexpected results that I'm hoping you can try to interpret.
Thank you.
Thank you.
No, thank you very much for really interesting talk. So my question is, how about the climbing fiber synapses? Do they change during sleep?
We haven't studied them. It would be nice to, we, as I mentioned, we actually specifically try to avoid them for now. to focus on the parallel, and that's why we really sample the most distal part of the branchlet where there are very few. And so we will have to basically acquire new stacks and trying to be, but the problem for us is that if you go in more, where there is a mixture, it's not obvious, at least based on electron microscopy, how would you be able to distinguish between parallel and climbing, except for the overall size. And so, but the answer is that we don't know because we haven't done it. And I know that also there, the view is changing, that, they can be also very plastic, maybe in a different way. We just have not done any study on them.
Okay, thank you.
Thank you so much, and thank you everybody. That was just a beautiful presentation. I'm so grateful for the time that you spent with us. I hope you have a wonderful Tuesday. Thank you, everyone.
Thank you. Thank you again for the opportunity. Bye.
Bye.